So this is chapter 15. This is actually a brief section of just chapter 15, but I wanted to make sure you had this on video so you could look back to it more regularly. So this is protein synthesis. Now protein synthesis is from DNA to protein, and in protein synthesis the basic idea is that, remember, DNA is the genetic code. It determines who you are and what you do. But from DNA, it has to go into another format of that language, and then it has to then turn into a completely different language. So this protein synthesis is a multi-stepped process. So this right here, this is hemoglobin. If you remember back, hemoglobin is a protein. It's actually a 4-component protein. This would be then the quaternary structure. There's a tertiary structure, another tertiary, another, and another. And remember, this is what carries the oxygen in your blood. This protein, this specific structure, is all determined by DNA. DNA is what determines what the structure of the protein is going to be, so the structure of that hemoglobin is determined by the initial DNA. Now, this is what's known as the central dogma of life. And so what's that really known for, or why it's known as central dogma, is because it's this idea that again, if you take DNA, and DNA, you remember, is in both eukaryotes and prokaryotes. Prokaryotes are just not in a specific nucleus, whereas eukaryotes it is. But that DNA eventually is that strand of DNA. After a process called transcription, that DNA then becomes RNA, and then after a process called translation, that RNA becomes the protein that we need to be used, either as an enzyme or as a structural component or as a signaling or some kind of communication form. So this is what's known as, again, this is known as the central dogma of life. And so it's a whole process. So again, it's known as the central dogma. And what that is DNA to RNA to protein. And I don't know if you know what the Rosetta Stone is, but basically what the Rosetta Stone was it was a stone that had 3 languages on it. And so for a while there, we didn't understand what's called cuneiform, which is one of the first original written languages. And the Rosetta Stone allowed us to use a different set of languages to find an old language that we didn't know how to say at all. So it's this way, is that the language of DNA, with its G's, A's, T's, and C's, is different than the language of RNA. And the language of RNA is different than the language of proteins, because if you see, RNA now has a U, or a uracil nucleotide, and proteins aren't made of nucleotides, they're actually made of amino acids. So you had to have some form of translation between each of these languages, and RNA is that translation piece. So you transcribe, or transcribe means to write, so you're scribing into a new language. That language then is able to speak into this. So it's kind of like if you knew French and English but you didn't know Spanish, but you need to talk to someone that spoke Spanish, but this person knew both French and Spanish, you could speak English, you could speak French to this person, and this person could speak Spanish to that person. So there's a translation, and that translation from a DNA to RNA to protein is known as central dogma. So I do want you to kind of know, though, that there are differences between the protein synthesis of a prokaryote and of a eukaryote. Mainly 2 main things. First off is location. So the first difference is location. Now, the location is because on a prokaryote, the location is basically everywhere. It's in the cytoplasm, because remember, the prokaryotes do not have a nuclear bound membrane, and so there is no transfer. Secondly is a post-transcriptional post-translational processing. So not only does it not just occur just all over the place, it also does not have a post-translational processing, whereas eukaryotes do. So those are the main 2 things. First off, the location. On the location, protein synthesis, transcription, and translation all occur in the cytoplasm. So very simply, it can take the DNA, and very simply take that DNA, change it into RNA, and that RNA can then be changed into a protein. Okay? And that all occurs in the cytoplasm. Whereas on the eukaryotic side, remember in the eukaryotic side we have a cell, and that cell has a nucleus. That nucleus contains the DNA. But the ribosomes, again, the ribosomes is where this RNA is going to go to become proteins. Those ribosomes on the outside of the nucleus. So the DNA has to go outside and the DNA has to change into RNA, and then RNA is then going to transfer outside of the nucleus and then go into the ribosomes, and those ribosomes are then going to what's going to make the protein. Okay? So first off, the location, it only occurs in the cytoplasm, where it occurs in two different locations in the eukaryotes, both the nucleus and the cytoplasm, or the ribosomes. And then secondly, post-transitional. I'll talk about this more specifically later, but the post-translational processing is because since the RNA has to transfer a greater distance, there's greater chance for there being loss or changes to the RNA. So there's different ways that they protect that RNA. So first off, they're all being initiated, and here's actually the eukaryotic initiation of DNA. I'm sorry, of protein synthesis. If you look, the first one is what's called the TATA box, T-A-T-A. And so it's basically a sequence, and it literally says T-A-T-A, thiamine, adenine, thiamine, adenine. It's a box, and then it's basically the box is essentially the go box. This transcription factor once it finds that go box, other transcription factors attached to it. And this is what's known as the initiator. When that initiator is formatted, the RNA polymerase comes along. The RNA polymerase then is what makes more polymerase, or more RNA. This whole complex, the RNA polymerase and the initiation factor, the transcription factor, all become what's known as the initiation complex. And from that initiation complex is where the mRNA is formed. Okay, so it has to have this transcription factor. It has to have this transcription factor on the TATA box for it to continue the initiation. The initiation is then attached to the RNA polymerase. The RNA polymerase then starts to build the new RNA that's going to come out from the DNA. So these are eukaryotic processing. Like I said, the eukaryotic processing has to basically do two different things. First off, well, technically it's more three, because I said two first off, because the first thing is it adds a tail and a cap. Okay, so the tail is on the 3' and the cap is on the 5' side. The tail is a poly A tail, so all it is a bunch of A's all lined up at the very end. And so what that does, that protects the very end. Remember, this has to transfer from the nucleus through the cytoplasm into the ribosomes. So there's a chance that could get cut off, that could get lost, those kind of things. So the poly A tail protects it. And on the very other end, it does what's called a G' cap. And so what it does actually adds a large complex of proteins. If you look down here on the bottom, a methyl group of guanine phosphates down here on the end. Again, that also allows for protection. So the first step is a tail and a cap, and the next thing it has to do is it has to remove the introns. So again, this is all occurring before it leaves the nucleus. And the introns are just sections of DNA that are nonsense DNA. So if you look at this picture, the introns are labeled in purple. Okay, so those introns are going to be removed, and notice after the introns are removed, the RNA section is actually much smaller. Since the RNA section is much smaller, that then leaves the nucleus. So not until the introns are removed, and it's only the exons exit the nucleus, and so not until those are all removed can the exons and the full mRNA leave. So this is what's called post-transcriptional RNA. So once it gets out of the nucleus, it has to go to the ribosome, and the ribosome again is the factory. That's where the proteins are produced. You notice there's actually 4 different sections within the ribosome. and it's actually the P, the A, and the E. And again, the only vocabulary word that's from here is the A site, and the A site is actually the active site. That's actually where the protein is being made. So essentially, what you have here is right here is the RNA. The mRNA is going into the ribosome. If you zoom in right over here, notice that the RNA is made of a bunch of triplet codes, and those triplet codes is the reading, the specific words that determine what amino acids need to come. And those amino acids join together, you look up here, those amino acids join together and form this polypeptide. And that polypeptide is the new protein. Okay? And so if you notice these, you notice that they are in 3-layer codes, and so we actually read those 3 layers at a time. That's what's called a codon. And the anticodon is actually what's attached to the tRNA. And the tRNA is that kind of yellow globular area right up here on the top. The tRNA brings the amino acids over and attaches to the mRNA, and that's what determines what amino acid is based on the codon, and the anticodon is on the tRNA. And so then there's another one available, and there's another one. And so again, it's the active site, the P site, and then the E side. The E side is actually where it's ejected or leaves. So here's the whole process on a eukaryotic cell. So again, the DNA goes through transcription, and that transcription then forms RNA. The mRNA is processed. Remember, it has a 5' cap and a 3' poly A tail. It also removes the introns, and those introns then are removed and reused later, but then the exons and the 5' cap and the 3 poly A tail go out of the nucleus. The nucleus then, the mRNA then transfers into the ribosome complex. The ribosome complex then feeds the RNA through, and 3 by 3 it reads that code, and that code will determine what protein or what amino acid needs to be brought over, and those amino acids then link together through peptide bonds to form protein. So here's the codon chart. You need to be able to read this. This codon chart is hopefully you remember from freshman year. But the codon chart is essentially you read the first, the second, and then the third letters. So let's say, for instance, you have an A, C. You read A. The first letter is A, second letter is A, third letter is C. So you say, okay, it's got to be on this row, in this column, and you say, okay, well, C, AAC right there. So AAC is asparagine. So you look again, you say, okay, well, what is UGG? So again, you say, okay, it's got to be in the U column. It's got to be in the G column there. And the third letter is G, so it's got to be UGG. And UGG is tryptophan. Now, sometimes they're not labeled this way where exactly it is. It's labeled other ways, but it's the same process every single time. You start on the left side, you go to the top and the right. It's always labeled that way. And again, I mean, you can do all day long. You can do, okay, well, what's GGG? You say, okay, well, here's G. Here's G, and I actually literally take my 2 fingers and I bring them down and say, okay, well, it's GGG, and so, okay, say, I know I'm in this section, well, GGG is right there. So then I know what it is, it's glycine. So you need to get comfortable with the codon chart. I'll give you some activities, those kind of things to use later. Now, I want to erase all this because I want you to be able to come back to this here at the end of the video and use this codon chart, or you can look up your own codon chart online. They're pretty available. Just type in codon chart, because I want you to practice on this last section, this last slide. Alright, so there's your chart. So here's the whole process right here. Okay, so again, we've got transcription. Transcription transcribes into an RNA. The RNA then goes outside of the nucleus through the cytoplasm into the ribosome. The ribosome then feeds the mRNA through 3 layers at a time. That then connects to this. Okay. So what I want you to do is I'm going to open up a new screen. And to bring back to class, or this is what I want you to submit on your form below. Let's say if your DNA code is A, G, C, A, G, C, A. Okay, so if your DNA code is that, and I'm going to mark my introns. Now this is just random, so it's just not always this way. So here's my introns. What I want you to do is finish up when I end. So, okay, so if DNA is that, then the RNA has got to be U, U, C, G, U, C, G, U. Okay, because again, remember, A is to T is on DNA, but A is to U on RNA, because the uracil is around the thymine. Okay, now, that's our pre-mRNA. Well, our post-mRNA has the 5' cap, the G' cap. It also has the introns removed, so we don't need that one. And we don't need that one, because again, those are introns, so these are all exons. Now, of course, there would be more here, because it's not enough for the 3-letter cuts. And then at the very end, there's going to be the poly A tail. So what I want you to do is tell me what amino acid sequence, or what amino acid should be right there. So what I want you to do is fill in on the form, go back to this slide. So go back to this slide, and I'll go ahead and go back to the codon chart, and tell me what amino acid will U represent. Again, make sure that you complete your whisk on a separate sheet of paper. We'll turn those in next time in class. We'll also go over those as a group work. So again, to summarize, you're going to summarize what you learned, something new or something that was reinforced, and then also make sure you complete a question, either a confusion, a discussion, or an example question. Also on the attached form down below, complete this one question: what does U going to represent on the codon chart? What amino acid? And you can just give me the 3-letter code.